This is part 3 of 3 on chapter 11. So in this chapter we're looking at physical and chemical agents for microbial control. So in the first two parts we kind of went through some terms. The second part we looked at the physical agents, so the heat and the UV, or the radiation part, the different types of radiation. So this one we're going to be looking at the chemical agents and then a little bit at mechanical agents. So just to remind you, this is what we're looking at. So in part 2 we looked at these pink types of agents, so the physical agents, like I just said, the heat and the radiation. This part we're going to be looking at chemical agents and then also the mechanical removal methods. So we'll just look at filtration very quickly. Alright, so first to get filtration out of the way. So filtration, we're just physically removing microorganisms, and we do this by passing gas or liquid through a filter. So basically you need a filter that the holes are small enough for the gas or liquid to fit through, but it stops any of the bacteria or other organisms from passing through. So in this example we have water. We're going to filter the water. So water molecules are very, very tiny compared to bacteria cells. So the pores are extremely tiny, liquid fits through, bacteria cells get trapped. And filtration, it's used to sterilize heat-sensitive liquids. It's used to sterilize air in hospital isolation units, or to sterilize air in industrial clean rooms. So if you're creating like computer components, you don't want any dust getting in there or microorganisms. The hospital isolation units, they use the HEPA filters in order to get everything out of the air. Alright, so that was the filtration. So we're going to be looking the rest of the time at the chemical agents. So chemical agents, you can have disinfectants, you can have antiseptics, you can have sterilants, degermers, and preservatives. So chemicals are used a lot to control microorganisms. So qualities that we look for in chemicals, we want rapid action at low concentration of the chemical, so we don't want to use a lot of the chemical to get good results. It's better if the chemical we're using is soluble in water or alcohol, which makes it a little more stable, it's easier to work with. Chemicals, we want them to be broad spectrum, which means that we want the chemicals to kill a wide range of microorganisms. But we also want it to have low toxicity to humans or other animals. We want the chemical to be penetrating, we want it to work really well. We don't want it to be corrosive or staining, so we want to be able to use it on equipment, like medical equipment. And the chemical also has to be affordable and readily available, so it has to be easy to get a hold of, and also at a decent price. Because hospitals and veterinary clinics, they have to run like a business, but so you have to get these affordable things. There are different levels that chemicals will work at. So some chemicals are high-level germicides, where they're going to kill those highly resistant organisms like the endospores, for example, or the viruses. So some of these germicides, germicides means cidal is to kill, so germicides, killing germs. So some of these germicides are sterilants. They're going to sterilize the object that they're used on. So these high-level germicides, are going to use them on devices that are not heat sterilizable, so we can't put them in an autoclave, and devices that are intended to be used in sterile environments, such as body tissue. So things like instruments used in surgery, you want to make sure that they're sterile, that there's no endospores, there's no viruses on these instruments. The intermediate level germicides, these are going to kill those moderate resistant things like fungal spores, but they don't kill the endospores. They kill bacillus, tuberculosis bacillus. They can also kill viruses. So these germicides, the intermediate level ones, they're used to disinfect devices that are going to come in contact with mucous membranes, but they're not invasive. So You're not going to be using these on instruments that are used in surgery where you're actually cutting people open and getting in there, but you can use them on things that touch the mucous membranes, like inside the nose, for example. And then there's low-level chemicals or germicides. They eliminate only the vegetative bacteria or vegetative fungal cells. They can kill some of the envelope viruses, so the things that have the low resistance. So these chemicals, they can be used to clean surfaces that touch skin, so the outside, but you don't want to use them on things that are going to touch the mucous membrane, because then you can still get fungal spores into the membranes. Okay, so germicidal categories, there's 10 different categories, and I'm going to quickly go through them. You don't have to know exactly them, like the halogens. This kind of relates to organic chemistry, so if you haven't had organic chemistry, it's kind of hard to explain the halogens or the phenolic compounds or the alcohols. But just kind of generally know what they are. So halogens include the chlorine or the hypochlorites, so this includes bleach. The bleach works to denature proteins, so it basically breaks apart proteins and they don't work anymore. It's an intermediate level type of chemical. We use chlorine bleach in water, sewage, wastewater. We use it to clean inanimate objects. Another type of halogen is iodine. This also denatures protein. It's an intermediate level. It's more mild than bleach. Bleach is very, very hard on the skin, and it also smells really bad if you've been around it. Iodine, it's a little milder for medical and dental. They use iodine to disinfect the skin before giving you vaccinations or other types of shots. They can put iodine into ointments that are used on the skin. So iodine is used a lot in the medical field. Phenolics, these chemicals work to disrupt cell walls and membranes. So they make the cell walls break apart or make the membranes break apart. They can also make proteins no longer work. Phenolics are usually low to intermediate level. One example, so triclosan, that's an ingredient in antibiotic soap. So if you actually look at the soap in the lab, it has this triclosan in it. Lysol is another type of phenolic compound that's used to disinfect areas. So these are disinfectants. They are used to get rid of microorganisms that could potentially cause disease. But they don't sterilize the area, so they don't get rid of everything. Another type of phenolic compound is chlorhexidine. This is a surfactant, so it breaks apart cell membranes and it denatures proteins. It's broad spectrum, so it gets rid of a lot of microorganisms. It's low to intermediate level. This one is used as a skin degerming agent. It's used in preoperative scrubs. It's used to clean skin. It's used for burns. And this one I'm pointing out because it can be used to control MRSA, which is a type of Staphylococcus bacteria that is resistant to a group of antibiotics. So MRSA is becoming a bigger and bigger problem in hospitals because it's getting hard to kill it. It's basically a type of bacteria that has evolved antibiotic resistance. And then also Actinobacter outbreaks. This is another type of bacteria that causes problems in clinical environments. Alcohols, ethyl alcohol, isopropyl alcohol, so rubbing alcohol. These act as surfactants, so they dissolve the membrane, the cell membrane, which leads to cell death, and they work at the intermediate level. Hydrogen peroxide. Hydrogen peroxide's a really good antiseptic if it's used at the 3% level. So hydrogen peroxide, it produces really highly reactive free radicals, which basically are really dangerous to proteins and DNA. These free radicals, they just bounce around destroying DNA, and they also decompose to oxygen gas, which is toxic to anaerobic bacteria, so bacteria that cannot be in an environment with oxygen. So hydrogen peroxide is good in that way. Like I mentioned, it's an antiseptic at really low concentration, so the hydrogen peroxide you buy at the store, the 3%, you can use to clean wounds. And the stronger solutions are sporicidal, so they can be used to sterilize different types of equipment that may be sensitive to autoclaving, so like the high pressure, so sensitive to heat or other methods. Aldehydes, these kill microorganisms by destroying proteins and DNA. So glutaraldehyde, it's a 2% solution of this. It's called Sidex. It's used to sterilize heat-sensitive instruments that cannot go into the autoclave or use other types of heat methods. There's also formaldehyde, which can be used as a disinfectant, or we use it as a preservative. However, it's really toxic, which they figured out, so we don't really use formaldehyde very much. Even for preserving tissues, they are getting more and more away from the formaldehyde. Gases and aerosols, so there's different types of gases that you can use. It's kind of like an autoclave, but instead of pumping in water in there, and you pump in these different gases. So ethylene oxide, propylene oxide, they're really strong alkylating agents, so basically they just destroy the cells. They work at a really high level, so they can be used to sterilize or disinfect plastics, prepackaged devices. Sometimes we can use it for food. Detergents and soaps. Soaps are used to mechanically remove soil and grease that contain microorganisms. So some soaps are non-germicidal. So non-germicidal soaps are just used to reduce the number of microbes on the surface. Germicidal soaps that have other chemicals in them, they actually reduce the number of microorganisms. Another type, heavy metals. So things like silver and mercury actually kill bacteria cells when they're in really low concentrations. And silver and mercury, they inactivate proteins by binding onto the proteins, and then it causes the proteins to no longer work. They work at really low levels. So things like silver or gold or mercury, they can inhibit cell growth on certain things. The ninth type of chemical agent are dyes. So there's aniline dye, which is used against gram-positive species of bacteria and some fungi. Aniline dye can be used on skin, so it's a type of antiseptic, and it can also be used in wound treatment. It's a very low-level antimicrobial agent, and it has a narrow spectrum because it only works on certain types of bacteria, so the gram-positive bacteria. There's acids and alkalines. So acids and basic, these usually have really low level of activity. So we have organic acids prevent spore germination, bacterial and fungal growth. Acetic acid inhibits bacteria growth, which you see acetic acid in a lot of food items. It's used to preserve the food items. Proponic acid prevents mold growth. Lactic acid prevents anaerobic bacteria growth. So anaerobic, remember, is without oxygen, so bacteria that grow in environments without oxygen. Benzoic and sorbic acid, these are other acids you find a lot in food. These inhibit the growth of yeast. So a lot of these are actually used to preserve food, so like crackers or other types of food that you put on a shelf, to prevent it from going bad, from spoiling. So this concludes chapter 11. We looked at lots of different ways to control microbial growth, including physical agents. We looked at mechanical agents, so filtration in this part, and then the different types of chemical agents. So this kind of summarizes these different types of agents. And remember, in the first part we looked at the different circumstances or the different questions you need to look at to decide what type of method you should use For example, cleaning skin, or if you have an instrument that can't go in the autoclave. Those are things you'll need to look at for when you're picking out a method to use.